Okay. COVID-19. Do you remember that? You guys remember COVID-19? Coronavirus disease 2019. That's why it was called COVID-19. Did you guys know that? Is a contagious disease caused by the coronavirus SARS-CoV-2. A coronavirus, they are a group of related RNA viruses that cause diseases in mammals and birds. In humans and birds, they cause respiratory tract infections that can range from mild to lethal. And then SARS-CoV-2 stands for severe, meaning the symptoms are extreme, you can't ignore them, right? Acute, it's short-term, lasting. Respiratory, meaning it affects your lungs, your respiratory tract. Syndrome is what the disease is. And then it's caused by the, I think, coronavirus 2 strain, COVID-2, I think, or whatever. Or no, I think this might be the name given to the whole, no, this is the name given to the whole strain, I think, of the virus itself or whatever. But, okay. In January 2020, the disease spread worldwide, resulting in the COVID-19 pandemic. The symptoms of COVID-19 can vary, but often include fever, fatigue, cough. Don't care about that. Don't care about this, or that. The first known case was identified in Wuhan, China, in December of 2019. Coronavirus 2. Wait. Hold on. COVID-19 is a disease caused. I'm in extreme disagreements. I'm a Texas cop, and Darius is going to fuck around and get himself in much bigger trouble because I'm essentially violating the conditions of his bond. What? Hold on. Why are you messaging me? Why not message Darius? Hi, what's up?
Destiny, real quick.
Yeah, go.
Yeah, so hold on, let me mute stream. Okay, so in Texas, Travis County is, I believe, where Darius lives because he's outside of Austin. No doxing. If he was arrested for assault BI family, which is what y'all listed, he's almost certainly going to have a condition of bond either to not contact the victim, meaning in no way, don't talk to him. Excuse me.
Mm-hmm.
Don't talk to him. Don't send him a letter, text, in their chat, nothing, right?
Well, there's certainly Darius would know what the conditions of his bond are. He would never be.
Hey, retard, why wouldn't you just message me? Why are you going to message Destiny about a pseudo problem, dipshit?
Did you check your conditions of bond, Darius?
No, did you check my Discord and message me about an issue you had, dumb fuck? Or do you just want to talk to daddy?
Did you check your conditions of bond?
No, I'm going to answer my question, I'll answer yours.
I don't fuck around on Discord, dude. I don't.
Oh, we can tell. Oh, yeah, by you messaging Destiny, you don't fuck around on Discord, my ass, nigga.
To be heard.
And I did check my conditions of my bonds, and I talked to them already. Yes.
Okay.
What
all right. I just wanted to make sure, man, because that's not up to the victim. That's
you didn't want to make sure. You wanted to talk to Destiny and get on stream, you fucking loser. Neck.
All right. You have a good day, Darius. Just wanted to make
you too,
sure
dumb fuck.
Okay. Based? Wait, sorry.
Base?
Are we in July? The 7th month is July, right? Okay. If I recall correctly, there was a paper that was published initially that I don't believe has ever been retracted. probably look that up. And we'll give that guy a read. We'll see what's listed here. Fuck, I don't remember which sources. I don't care that much. This is by Scott Alexander, right? Sar Wilf is an ex-Israeli entrepreneur. Since 2016, he's been developing a new form of reasoning meant to transcend normal human bias. His method, called Rootclaim, uses Bayesian reasoning, a branch of math that explains the right way to weigh evidence. This isn't exactly new. Everyone supports Bayesian reasoning. The statisticians support it. I support it. Nate Silver wrote a whole book supporting it. But the joke goes that you do Bayesian reasoning by doing normal reasoning while muttering Bayes, Bayes, Bayes under your breath. Nobody, not the statisticians, not Nate Silver, and certainly not me, tries to do full Bayesian reasoning on fuzzy real-world problems. They'd be too hard to model. You'd make some philosophical mistake converting the situation into numbers and then end up much worse off than if you tried normal human intuition. Rootclaim spent years working on this problem until they were satisfied their method could avoid these kinds of pitfalls. Then they started posting analyses of different open problems to their site, rootclaim.com. Here are 3. Holy shit, they go hard. I'm trying to think if I want to read this first or the paper first. For example, does Putin have cancer? Or here are some. Was there widespread fraud in the 2020 election? And then the calculated results of their claims. Does vitamin D reduce the severity of COVID-19 outcomes? Does Putin have cancer? Okay. For example, does Putin have cancer? We start with the prior for Russian men, ages 60 to 69, having cancer is 14.32%. According to health data, we just for Putin's healthy lifestyle, minus 30% cancer risk, and lack of family history, negative 5%. Putin has advantage from the world stage for long periods of time, which seems about 4 times more likely to be true. If he didn't have cancer than if he did, about half of cancer patients lose their hair, and Putin hasn't, so we'll divide by 2. On the other hand, Putin's face has gotten more swollen recently, which happens about 6 times more often to cancer patients than to others, so we'll multiply by 6, and so on and so forth, until we end up with the final calculation. 86% chance Putin doesn't have cancer. Too bad. This is an unusual way to do things, but Sar claimed some early victories. For example, in a celebrity Israeli murder case, Sar used Rootclaim to determine the main suspect was likely innocent and a local mental patient had committed the crime. Later, new DNA evidence seemed to back him up. One other important fact about Sar, he is very rich. In 2008, he sold his fraud detection startup to PayPal for $169 million. Since then, he's founded more companies, made more good investments, and won hundreds of thousands of dollars in professional poker. So, in the grand tradition of very rich people who think they've invented new forms of reasoning, Sar issued a monetary challenge. If you disagree with any of his Rootclaim analyses, you think Putin does have cancer or whatever, he and the Rootclaim team will bet you $100,000 that they're right. If the answer will come out eventually, for example, wait to see when Putin dies, you can wait and see. Otherwise, he'll accept all comers in video debates in front of a mutually agreeable panel of judges. Since then, Sar and his $100,000 offer have been a fixture of internet debates everywhere. Is he betting this? This is like a legitimate two-sided bet, right? Like, you would have to put your $100,000 up against his. It's not just like a $100,000 offer to beat a system, right? I'm guessing. Since then, Sar and his $100,000 offer have been a fixture of internet debates everywhere. When I argued that vitamin D didn't help fight COVID, people urged me to bet against Sar, and we had a good discussion before finally failing to agree on terms. When anti-vaccine multimillionaire Steve Kirsch made a similar offer, Sar took him up on it, although they've been bogged down in judge selection for the past year. Rootclaim also found in favor of the lab leak hypothesis of COVID, When SAR talked about this on an old AX or ACX comment thread, fellow commenter TGOF137, Peter Miller, agreed to take him up on his $100,000 bet. Let's see, what's this guy's name? SAR Wilf is an ex-Israeli entrepreneur, multi-millionaire who claims to have invented a system of analysis called a Rootclaim, where he takes bets against predictions made by his system. Peter Miller took him up on a debate challenge 2024 concerning his prediction that COVID-19 originated in a lab. Oh, Astral Codex 10 from his own website. Nice. You were completely right on this issue. Years of dealing with similar controversies in Rootclaim taught me that truth is hard. We are constantly surprised with the results of our analyses, and each time find a different party to be correct. Sometimes it's US, sometimes Russia, sometimes China, sometimes Democrats, sometimes Republicans, sometimes the mainstream, sometimes the fringe, sometimes experts, sometimes lay people. In the case of Kavanaugh, it's easy to see how his simplistic approach leads him to wrong conclusions. He takes the mainstream opinion on the chemical attacks of Syria. Our analysis originally yielded 92% for the opposition carrying out the good attack. And that was recently shown to be the case after a video of opposition fighters launching chemical rockets was geolocated to the attack's launch location. He does the same for the origins of SARS-CoV-2, where the evidence strongly favors lab leak. There are no shortcuts to truth. I disagree and think that the lab leak theory of COVID's origin is unlikely to be true. I say this having previously endorsed the lab leak theory, or at least having said that the question was a toss-up. After taking the time to dive into the issue, I now think it's highly unlikely that SARS-CoV-2 was created in a lab. I first heard of the Rootclaim challenge a few days ago when Scott mentioned your debate with Kirsch. Would I debate the lab leak theory for $100,000? I don't know. That's a large quantity of money for me, so I needed to know exactly how the debate would be judged. Like, I think that Kirsch's claims about vaccine deaths are obviously fake or false. In a fair contest, I'd bet all the money I have that COVID vaccines have saved more lives than they've cost, but I wouldn't debate that with Kirsch because I don't think the rules of his contest would be fair. In the case of your debate, it looks like he wants to rig the judges to be in his favor, i.e. by nominating Weinstein and Martinson as judges, or at least neutral on the question, the 6-to-8 balanced judges idea, so that the debate will end up inconclusive. I've been thinking about how to frame these debates in a way that could be objectively decided. Like, I don't think it's fair for you to compare the Rootclaim challenge to James Randi's $1 million prize. For psychics to enter Randi's prize, they don't have to put up $1 million of their own. That's what I was comparing it to. Randy never, you don't have to put your own money up there. And the outcome of each contest could be objectively judged. Anyone could watch and see whether or not the paranormal powers were working. Explain why that's funny? Because Weinstein is a fucking lunatic. I think some controversial questions about COVID could be objectively answered, like I think we can objectively test the question if masks work with a simple challenge trial. The lab leak theory is harder to decide without some form of judging. Here's one bet I would propose. No one will ever prove the lab leak theory. The bet needs some time frame to resolve because never is a long time. I might frame it as in 5 years' time, the scientific consensus will not be that SARS-CoV-2 was created by gain-of-function research at the Wuhan Institute of Virology. We can define consensus in a variety of ways. I mean, something comparable to, say, the level of scientific consensus that global warming is real and mostly caused by human CO2 emissions. It could be something like surveying if 90% or more of virologists believe the lab leak theory. It could be done by nominating current zoonotic origin proponents and saying they won't endorse the lab leak theory. I might suggest people like Michael Worobey, Bob Gary, and Christian Anderson. Two of those scientists already changed their minds from lab leak to zoonotic origin in the early 2020. So that should show that they are capable of changing their minds. Worobey also signed a letter endorsing the discussion of the lab leak as a valid question, even though he strongly advocates for zoonotic origin. I would bet that the lab leak discussion will remain about the same as it is today. Most virologists will still lean towards a zoonotic origin. Scientists will continue to slowly find somewhat related bat viruses in China and Southeast Asia. The lab leak theory may well live on in the public imagination. There will still be podcasts and Substack articles and books written about the Wuhan lab. The theory might be endorsed by more government reports, like the House GOP Minority Report, which already endorsed the lab leak theory, and may even become the theory advanced by the majority of the U.S. government if the GOP wins the next election. But I would bet that the lab leak theory will still remain fringe among scientists. There will not be any clear proof of a gain-of-function research program at the WIV creating the virus. No one will find proof of the RNA sequence that COVID is not natural. Okay. And so they agreed to the debate here. This was in 2023, February. At the time, I had no idea who Peter was. I kind of still don't. He's not internet famous. He describes himself as a physics student, programmer, and mountaineer who obsessively researches random topics. After a family member got into lab leak a few years ago, we started investigating. Although he started somewhere between neutral and positive towards the hypothesis, he ended up 90% plus convinced it was false. He also ended up annoyed. Contrarian bloggers were raking in Substack cash by promoting lab leak, but there seemed to be no incentive to defend zoonosis. Zoonosis or zoonosis? Zoonosis, right? Unlike Tsar, Peter was not especially rich. 100K represented a big fraction of his net worth, but he wrote me in an email. It was a moderately large financial risk for me. I expected a smart and unbiased person would vote for zoonosis with, say, 80% odds after seeing all the evidence. If both judges voting for lab origin is uncorrelated, that's 20% squared, and it was pretty low odds of a catastrophic financial risk for me. I wasn't highly worried about losing the debate because I was wrong about the science. I put in enough to know I'm probably correct there. My biggest fear was that I'd choke at the debate for some reason, that I'd be too anxious, and particularly that I'd be unable to sleep the night beforehand. I have zero prior debate experience to rely upon. If this seems like a weirdly blasé attitude towards risk, Peter told blogger Philip Marcolin that he is a mountain climber, or sometimes there is a 5% chance to die, and the stakes are just not that high for a debate. Unlike the eternally bogged down Tsar-Kirsch debate, here things moved quickly. The two contestants put out a call for judges on the ACX subreddit and agreed on Will von Truren, a pharmaceutical entrepreneur with a PhD from Stanford and a background in bacteriology and immunology, and Eric Stancifer, an applied mathematician with a PhD from MIT and experience in mathematical virology, both of whom received $5,000 as payment for their 100 hours of work, paid by the two contestants along with their $100,000 table stakes, with Scott taking $1,000 for escrowing it. The format would be 3 sessions, each consisting of hour-and-a-half arguments by both sides, and then 3 hours for the debaters to answer questions from the judges and each other. To the debate. Below, I've included the videos from each session, plus my long summary if you prefer text. In the second video on viral genetics, biotech entrepreneur and lab leak expert Yuri Dagin stood in for Tsar. Peter continued to represent himself. Okay. There was a paper in Nature, paper on origins of COVID-19 virus, I think. This is an overview. I'm pretty sure this paper was published in 2020. What is this? Is this what I'm looking for? Is this what I'm thinking of? April 2020. Oh, this might be it. Okay. To the proximal origin of SARS-CoV-2. To the editor. Since the first reports, I always thought the leak thing made sense, but the camp that trumpets it are crazy. Also, it seemed inconsequential in how we went about dealing with the Rona. Well, keep in mind that the United States, I believe, somebody can remember, but I believe our stated reason for leaving the World Health Organization from Donald Trump had to do with how he felt like they handled, like blaming China for the origins of COVID-19. I believe that was I think that was our stated reason for leaving the WHO. Since the first reports of novel pneumonia in Wuhan, Hubei province, China, there has been considerable discussion on the origin of the causative virus, SARS-CoV-2, also referred to as HCoV-19. Infections with SARS-CoV-2 are now widespread, and as of March 11th, 2020, 121,564 cases have been confirmed in more than 110 countries, with 4,373 deaths. SARS-CoV-2 is the seventh coronavirus known to infect humans. SARS-CoV, MERS-CoV, and SARS-CoV-2 can cause severe disease, whereas HKU1, NL63, OC43, and 229E are associated with mild symptoms. Here we review what can be deduced about the origin of SARS-CoV-2 from comparative analysis of genomic data. We offer a perspective on the notable features of the SARS-CoV-2 genome and discuss scenarios by which they could have arisen. Our analyses clearly show that SARS-CoV-2 is not a laboratory construct or a purposefully manipulated virus. Notable features of the SARS-CoV-2 genome. Our comparison of alpha and beta coronaviruses identifies two notable genomic features of SARS-CoV-2. Hold on. Alpha versus beta viruses? What's the difference here? Is this just has to do with like variants basically? I think so. Okay. Our comparison of alpha and beta coronaviruses identifies two notable genomic features of SARS-CoV-2. One, on the basis of structural studies and biochemical experiments, SARS-CoV-2 appears to be optimized for binding to the human receptor ACE2. And two, the spike protein of SARS-CoV-2 has a functional polybasic cleavage site at the S1 and S2 boundary through the insertion of 12 nucleotides, which additionally led to the predicted acquisition of 3 O-linked glycans around the site. Okay, I think cleavage site has to do with how it enters. It's the part of the virus that binds to the cell wall that injects the RNA into the cytoplasm, maybe? Let me check. Cleavage, that's not what I'm looking for. Furin is a protease. A protease is an enzyme that catalyzes proteolysis. Proteolysis is the breakdown of proteins into smaller polypeptides or amino acids. Protease. Is it protease? Okay. Okay. Furin is a protease, a proteolytic enzyme activated by substrate presentation. In molecular biology, a substrate presentation is a biological process that activates a protein. The protein is sequestered away from its substrate and then activated by release and exposure to its substrate. The substrate is typically the substance on which an enzyme acts, but can also be a protein surface to which a ligand binds. Ligand is an ion or molecule with a functional group that binds to etc. Okay. Okay, furin is a protease, a proteolytic enzyme activated by substrate presentation that in humans and other animals is encoded by the furin gene. Some proteins are inactive when they are first synthesized and must have sections removed in order to become active. Furin cleaves these sections and activates the proteins. It was named furin because it was in the upstream region of an oncogene known as FES. The gene was known as FUR for FES upstream region, and therefore the protein was named furin. Furin is also known as PACE, or paired basic amino acid cleaving enzyme. It is a member of family S8. Furin is a substilicin-like peptidase. Peptidase? Okay. Wait, where were we? What the fuck? Oh, I was reading this paper. Okay, sorry. Okay. The spike protein of SARS-CoV-2 has a functional polybasic furin cleavage site at the S1-S2 boundary through the insertion of 8 nucleotides. So this means that S1-S2 boundary is what? A boundary in the cell? And then how does furin work? Polybase. What is polybase? This doesn't look at all like what we're looking for. Okay, the power of polybase with SQL. With SQL, read local file. Okay, well, that's not what we're looking for. Okay. Cleavage.
So this is the spike protein genome.
Holy shit, we've got Kino audio.
This is the 13th video of the Prevent SARS-3 project video series. Our objective is to develop nanomaterials against these pandemics. So you can support this project. This video will focus on the putative engineered nature of the SARS-2 furin cleavage site. So this is the spike protein genome. This is a very critical capacity for SARS coronavirus 2 because only MERS has this capacity in previous human pathogenic coronavirus. As you can see, this insert the purple.
Oh, this guy's making the argument that it is artificial. SARS-2 clinical isolates after millions of infections do not have any recombination, indicating that the furin recombination. Okay, I just want an explanation what this is. Fuck. This is going to be like buried in some fucking molecular biology bullshit class that's never going to have.
The coronavirus has upended a lot of our lives. But understanding the biochemistry of the coronavirus can help us get out of the pandemic.
Thanks.
Especially understanding the protein, the spike protein,
Okay.
which is the protein that sticks off of the viral membrane, and it latches on to receptors on our cells called ACE2.
Okay, we got that part.
And then it gets cleaved by these protease, or these protein scissors. Then it undergoes this dramatic shape change that helps fuse the viral membrane with the cell membrane so the virus can dump its contents in and make the cells make more.
Okay.
And so today undergoes this dramatic shape change of the viral membrane, and it latches on to receptors on our cells called ACE2. And then it gets cleaved by these protease, or these protein scissors.
Where is the protease at? Is this on the, this is part of the virus, or does the cell provide this?
And it undergoes this dramatic shape change that helps fuse the viral membrane with the cell membrane so the virus can dump its contents in and make the cells make more.
It's in the cell.
And so today in part 2 of my
on
talk about
furin.
the spike protein, I want to tell you about the polybasic cleavage site.
Oh
aka
my
the furin
God.
Cleavage site.
Oh, we're getting everything. Okay, here we go.
The glycosylation, so like sugar chains
Yes.
that can stick off of the protein, and ACE2 binding and antibody binding. So
Okay.
let's take a look.
Let's go.
Setting the scene from yesterday's talk, I had told
Oh my God, we have S1 and S2?
you that the spike protein has these 2 subunits. The top
Okay.
part, like ahead of the broccoli, is the S1.
Oh, and these are on the spike protein. Okay.
The stalk is your S2.
Okay.
The
Wait, the
protease
stalk?
part, like ahead of the broccoli, is the S1. The stalk is your S2.
I don't know what a broccoli is. I don't know what the fuck that is. Okay. It sounds like some shit my parents wanted me to eat.
The protease cleavage cuts off the S1 subunit, which allows for the conformational change or the shape change. It exposes the hydrophobic, so a water avoiding region called the fusion peptide, then shoots up into the membrane and pulls it back in to fuse the membranes together. So the cleavage that needs to happen for that S1 to come off is this cleavage at this S2 prime site, so like right before the fusion peptide. But there's also this other site, this S1, S2 site. So it's
Okay.
called the polybasic site because basically, well, that
Is this like a whole separate day? Okay, damn.
wasn't supposed to be a pun. I know I make puns, but that one basic site because basically, well, that wasn't supposed to be a pun. I know I make puns, but that one wasn't supposed to be a pun. But a basic, so basic in terms of not acidic, so like the opposite of acidic.
Oh.
So something acidic is going to
It's gonna be Yo Mama.
donate a proton, so it's going to like give up an H plus. Something basic is going to take an H plus. When it takes an H plus, it becomes positively charged, so that's why basic things are often positively charged. And so there's these amino acids, so protein letters that can do this, and
Holy shit, I didn't even know that. What's the difference between an acid and a base? I didn't even know that. Or maybe I did at one point. Okay.
so in this polybasic cleavage site, so you have this RRAR.
Okay.
So R is arginine. It's one of these basic amino acids, and the reason, so that's why we call it a multi-basic cleavage site. And the reason we call it a furin cleavage site is because there's this protease, so a pair of protein scissors that likes to cut at these sites with lots of these basic residues, and so it can cut there. And so there was this big hoo-ha because the original SARS coronavirus doesn't have that site. This one does. Other coronaviruses also do have this one. It's not proof that it's engineered or anything like that.
Whoa! We're just coming here for information, buddy.
Especially because it's like not an ideal furin cleavage site anyway. So if someone was going to like engineer
Oh, you could make a better virus?
it, they would make a better one. So nothing like that. But that's what you'll see with like that S1, S2 thing. What else? So yes. So if you have it, that S1, S2, what can actually happen is that it can get cleaved while the protein's being made. So like during the processing step, like inside the cell before it even gets onto the surface. And so that can make it easier to cleave that second site. But remember, it can also make the protein like less stable and stuff. So it's not like slam dunk win. So that can make it easy. And then when you have like surface, you can have the early fusion at the surface. So that in that case, you have like a surface protease called temperase 2 that can cleave it at the surface. If not, you can have another protease called cathepsin cleave it inside. And so what we're talking about here, remember, is the cleavage at the S2 site, which is right before the fusion peptide. So that's the one you actually need to cut off the head of the broccoli. And so that what happens there is that you have this binding at the surface. It gets pulled in and enclosed in kind of like an intracellular quarantine called an endosome. So it's like this little pouch inside of the cell. Once it's in that pouch, the protease cathepsin can cleave it, and then that allows the protein to undergo this fusion process. So there are different ways that can happen. And so yeah, so here's just another view of the spike protein.
Oh, okay, I see now.
The S1, S2, and where the cut sites are. You'll also see that there's a lot of like little like weird things sticking off of the protein. Those are glycans. So those are like sugar chains. The protein, those are
A closer look at a single chain copy, protomer or protomes, is up and down configurations, or conformations. The whole purple ribbon part is the S1 subunit. It gets cleaved from the S2 subunit green somewhere back here. Okay, wait, do I have this so far? Okay, hold on. So cell, floating virus, when the spike protein plugs in, the cell provides the furin thing, and then that cleaves or cuts off the S1 part, and then the whole virus with the S2 part is then injected, and then there's a second cleave of the S2 part, and then I'm guessing then the RNA is injected into the cell from there, and that gets eaten by the ribosomes. Is that how this is working, basically?
Glycans. So those are like sugar chains. So sometimes you might hear a spike called a glycoprotein.
Okay.
That's a protein that has sugar chains attached to it.
Okay.
So sugar chains, we often think of sugar as like food and that sort of thing, but and like complex carbs, but sugar chains can actually be attached to all sorts of things, including proteins, including the spike protein.
Okay.
And so the spike protein, the glycans are helpful for the spike protein because it allows it to kind of like hide from our immune system. So basically, your body can make these things called antibodies. Your immune system makes these things called...
Wait, the shape changes when the furon cleavage occurs. Wait, so does cleavage mean it's not cut off, it just changes shape, or...?
Antibodies, which are little proteins that can bind to foreign things. And so it's like this random trial and error selection process to find antibodies that happen to match, to bind to the foreign...
Wait, sorry. When we make antibody drug conjugates, for example, modern cancer treatments, we often intentionally include a cathepsin B cleavage site so that after the conjugate is internalized, because the antibody bound something, the linker to drug is cleaved and the drug easily gets out of the endosome. Is that the pouch thing she was talking about? We can use this to target extremely cytotoxic drugs to cancer cells specifically. Okay. The cleavage induces a cut, and because of the thermodynamic nature of proteins, the whole thing then moves. Okay, a host enzyme like furin cleaves the spike protein at the S1, S2 site, separating S1, which stays attached to ACE2, which is the gate that lets the thing in, right, and S2. Another cleavage site at the S2 site by the host protease, like TMPRSS2, activates the S2 part. This triggers a shape change that lets the virus fuse its membrane with the cells. The viral RNA is then released into the cell. Oh, okay. So you get a cut at the S1 site, and then you get a shape change at the S2 site?
Thing and not to self proteins, and then the cells that make those antibodies, so the B cells that make those antibodies, they get selected for, and then they start making more and more of that antibody, and then they like keep some keep memory of it in your body so that if you see it again, you can combat the virus. And so these antibodies often bind to the same place as ACE2, and that can prevent the virus from getting into the cell. So if they bind to that receptor binding domain...
Wait, hold on. I'll just throw this video here, because why the fuck not? What am I looking for? Published date. Oh, July 28th.
So remember that was you can combat the virus. And so these more and more of that antibody, and then they like keep some keep memory of it in your body so that if you see it again, you can combat the virus. And so these antibodies often bind to the same place as ACE2, and that can prevent the virus from getting into the cell. So if they bind to that receptor binding domain, so remember that was that finger on the head of the broccoli. They bind there, then the coronavirus protein, the spike protein can't dock onto your cells, and then it can't get in. So if you have antibodies that can do that, we call them neutralizing antibodies. And so that's kind of like the goal of vaccines, is to induce these neutralizing antibodies that can bind to foreign to.
Wait, if you have the ACE2 gate on your cell, then I imagine there's probably some good stuff that gets into your cell based on that, whatever can interact with that. So how does your immune system know what the bad thing is and what the good thing is? Like, what if there was like a thing on your cell that's called like the nutrition gate, and a virus uses the nutrition gate to get in, and then your immune system creates a whole bunch of antibodies to make it so no nutrition gets into your cells? How does that work? Wait, sorry. I don't know if she'll explain it in that level of depth. I don't know if it matters to us. I'm just curious.
Bind to foreign things. And so it's like this random trial.
I wonder if she made this or if this comes from like a textbook. Antigen presenting cell ingests virus. Cell chops up virus's protein. Cell displays these protein pieces. Antibody making cells compete to find which make antibodies that bind to those viral pieces, but not the body's own molecules. Antigens are the things antibodies recognize and bind to. In this case, it would be the viral spike protein. Those winning antibodies makers get selected for, and more antibodies get made. These antibodies help the immune system find and destroy the virus in infected cells, and some stick around to keep watch, fighting less immunity. Vaccines and virus, the real infection part, introducing the okay.
Trial and error selection process to find antibodies that happen to match, to bind to the foreign thing and not to self proteins. And then the cells that make those antibodies, so the B cells that make those antibodies, they get selected for, and then they start making more and more of that antibody.
Oh, she's just explaining this. Okay.
And then they like keep some, keep memory of it in your body, so that if you see it again, you can combat the virus. And so these antibodies often bind to the same place as ACE2, and that can prevent the virus from getting into the cell. So if they bind to that receptor binding domain, so remember that was that finger on the head of the broccoli. They bind there, then the coronavirus protein, the spike protein can't dock onto your cells, and then it can't get in. So if you have antibodies that can do that, we call them neutralizing antibodies. And so that's kind of like the goal of vaccines, is to induce these neutralizing antibodies, which is why it's so important that we have that, we can stabilize that pre-fusion conformation of the spike, like we were talking about yesterday, by stabilizing that elbow part with the prolines, because we want it in the pre-fusion conformation, so our body learns to recognize the pre-fusion conformation, because that's the conformation where you have those receptor binding domains available to bind to the ACE2 protein. So this is showing the structure of the protein.
And you want the pre-fusion binding phase, because once it's in the fusion binding phase, the thing is already in the cell, and you're not going to be able to do anything with your immune system at that point, right? I'm guessing, is that she has a whole thing on her website where she does the pictures and everything. My God. By the viral proteins, such as antigens, trigger an immune response when encountered again. Antibodies can be made against any part of the virus. These viral parts that antibodies bind, typically a piece of proteins called antigens. Immature B cells have many viruses. Okay.
Bound to the ACE2 protein. So the ACE2 is the one on your cells, the spike is the one on the virus, and so they need to come together in order for the virus to dock onto your cells. And so neutralizing antibodies often bind to the RBD in the same place that ACE2 normally would, and that prevents antibodies to come together in order for the virus cells. The spike is the one on the virus, and so they need to come together in order for the virus to dock onto your cells. And so neutralizing antibodies often bind to the RBD in the same place that ACE2 normally would, and that prevents...
Oh, RBD is the connection site to ACE2 on the spike protein. Do we know what RBD stands for, or is that...
ACE2 from binding. And... so a lot of the mutations to the virus that people are worried about are ones that affect that RBD, affect that
receptor binding domain. Gotcha.
Location where ACE2 normally binds, because that can, that ACE2 normally binds because that's also the place, or that the mutations that people are worried about are the ones that are where the neutralizing antibodies bind, because those are ones that can prevent the
neutralizing antibodies from binding. So if they prevent the neutralizing antibodies from binding, but they don't prevent ACE2 from binding, then you have a bad combination, because then the neutralizing antibodies can't block the virus from getting in. Okay, I think that was all that I wanted to talk about. Oh, so one more thing. So that's with like active immunity, and so that's either with the vaccine induced or with like through infection you develop these neutralizing antibodies. And the thing with active immunity is that your body is making them yourself. Your body learns to make them yourself. So it's kind of like a teach a man to fish thing. There's also the give a man some fish thing. And that's called passive immunity. And so that's where you just give antibodies. So the body, you're not like giving the instructions for making the antibodies, and the cells don't know how to make the antibodies. You're just giving them pre-made antibodies. And so that's like the monoclonal antibodies and the cocktails of monoclonal antibodies. So the idea with the cocktails is that you have multiple antibodies, so that the virus, if it's resistant to one of them, it won't be resistant to both of them. And that way you can have a better chance of it working. And so monoclonal is just like, it's a single antibody, like lots of copies of this single antibody, but it's all the same antibody. With the polyclonal, like when your body like makes its own antibody response, it's going to be polyclonal. Like you'll have multiple antibodies that all bind the same protein, but in like different places or different, so we call it epitopes. But with the monoclonal, you have like a single one, and that's like, so if we go back to those cells doing that trial and error thing to try to find antibodies that bind, basically you have like, it would be a single cell gets selected and makes a bunch of antibodies. Then those antibodies, the process is like recreated in a lab, and so you have all the same antibody being made. But that's why we call it monoclonal, because it's like a clone from that single and original cell that made that one antibody, except here it's like artificially in a lab, but it's the same protein that you would normally make in your body. But in your body you would make this polyclonal response, so you have lots of different antibodies. There's also things called like T cells responses, which I'm not going to get into because I'm not an immunologist. I'm a biochemist. And so hopefully I got all of this right. I just wanted to give you a sense of like how to interpret things like this, because I like talking about things like broccoli and elbows and cartwheels and handstands, but I also want you to be able to keep up with the more serious scientific literature and stuff, and so be able to merge the two, which is like the goal of my life, kind of.
Wow.
So yeah.
Thanks.
Proteins being made. So like to donate a proton, so it's going to like give up an H plus. Something basic is going to take an H plus. So right is because there's this protease, so a pair of protein scissors that likes this big who is in.
There's I wish I could impart this. Okay. The way that she talks, it kind of seems I don't want to say dumb or unprofessional, but like it can kind of sound like that as opposed to somebody who's like giving kind of like a prepared or rehearsed speech, who's like reading down notes. Even if people are inarticulate, something that's worth, I think, paying attention to is if somebody sounds inarticulate because they haven't written down like a prepared speech or memorized a script or anything. When I see somebody who's talking through something and they don't have a rehearsed line and they're kind of like searching for a way to describe something, in my mind, this demonstrates a really thorough understanding of the material, I think in a really counterintuitive way. It's very annoying sometimes that somebody has like a prepared speech or like dialogue tree for something, people are like, oh, this guy really knows his shit. But then when they have to kind of speak off the cuff about something, they're completely and utterly incapable of understanding whatever the thing is they're talking about. This is why I hate people like Elon Musk and all these motherfuckers. Sorry, it's just a random thing. But maybe she is retarded. I don't know. She could be. Who knows? Okay. Sorry, hold on. One more quick thing. We have a video.
Coronavirus.
Video,
like many
3D.
viruses.
Not made by AI, because it's 3 years old as well.
Coronavirus. Like many viruses, it uses a protein on its surface to attach to and enter our cells. Antibodies that fit onto this protein can block the virus from attaching.
Got him.
Coronavirus mRNA vaccines teach our immune system to make these antibodies. How do mRNA vaccines do that?
Yikes.
mRNA is a genetic material that instructs our cells to produce proteins.
Wait, this is way more basic than what we were just doing, I think.
The mRNA in the vaccine is wrapped in a layer of fat particles called dendritic cells. Or fat mRNA in the vaccine is wrapped in a layer of fat particles that protect it and help it get taken up by specialized cells of the immune system called dendritic cells. Once inside these cells, the mRNA does not enter the cell's nucleus or interact with DNA, but remains in the cytoplasm with other mRNA molecules, waiting to create the enzymes our body needs. When ribosomes read the vaccine mRNA,
this is like a Parasite Eve CGI.
Pieces of the viral surface protein are made. These pieces are then displayed on the surface of the dendritic cell. The dendritic cell travels to a nearby lymph node, where it presents the surface proteins to other cells of the immune system. Some of these cells, called helper T cells, train B cells how to make antibodies that will fit perfectly onto the surface protein of the virus. Other cells stimulated by the protein pieces, called cytotoxic T cells, can kill virus-infected cells.
Wait, what is a dendritic cell? Real quick, dendritic cell are specialized immune cells that play. Oh, okay.
Now, when the coronavirus tries to infect us, our immune system is ready, immediately recognizing, neutralizing, and destroying it before we ever even have a chance to become sick.
Tch, that's what they say. Okay. 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 All right, where were we? The spike protein of SARS-CoV-2 has a functional polybasic cleavage site at the S1-S2 boundary through the insertion of 12 nucleotides, which additionally led to the predicted acquisition of 3 O-linked glycans around the site. A nucleotide is just 2 amino acids connected to each other, right? Or is it more than that? Or less than that? Nucleotides are organic molecules composed of a nitrogenous base, a pentose sugar, and a phosphate. The 4 nucleobases in DNA are guanine, adenine, cytosine, and thymine. A nucleobase, a 5-carbon sugar, and a phosphate group consisting of 1 to 3 phosphates. Okay. Okay, so nucleotides contain nucleobases, but it's not the same thing. Okay. Okay, which additionally led to the predicted acquisition of 3 O-linked glycans around the site. What does that mean? What is an O-linked glycan? Are a type of post-translational modification where sugar molecules glycans are attached to the oxygen atom of serine or it's processed as a glycan or distinct from glycans are attached to them. Okay, I don't think we have to worry about this. Okay. Wow, freaks, thanks for the 1 gifted membership. One, mutations in the receptor binding domain of SARS-CoV-2. The receptor binding domain, or the RBD, in the spike protein is the most variable part of the coronavirus genome. 6 RBD amino acids have been shown to be critical for binding to ACE2 receptors and for determining the host range of SARS-CoV-2-like viruses. Wait. What's the difference between an amino acid and a nucleobase? A protein, NBAs are DNA. Okay. Amino acids make proteins. Nucleotides make DNA/RNA. Oh, okay. Amino acid chains are what your ribosomes spit out when they read RNA? I think, right? Okay. Okay. These vids explain. Wow. Where were we? Okay, the receptor binding domain and the spike protein is the most variable part of the coronavirus genome. 6 RBD amino acids have been shown to be critical for binding to ACE2 receptors and for determining the host range of SARS-CoV-like viruses. Okay, so this means when we say 6 RBD amino acids, we're saying that there are 6 different amino acids that are part of the, I guess the S1 site is what binds to the ACE2 receptor. And depending upon which amino acids are there, that determines which cells it can enter for determining the host range, I think, right? I have 2 biochemists in chat. I'm relying on your... Yes. Okay. That guy said yes. All right. Okay. With coordinates based on SARS-CoV, they are Y442. Oh, host ranges might mean species, is it? Okay. My guess was types of cells, but maybe that's, as we look at the source. 7 is... which regulate both the cross-species and human-human transmissions of... Oh, okay, so it is species. Host is okay. Okay, all right. Okay, with coordinates based on SARS-CoV-2, they are, or SARS-CoV, they are Y442, L472, N479, D480, T487, and Y4911, which corresponds to L455, F486, Q493, S494, N501, and blah, blah, blah. Okay. Are these coordinates, are these like coordinates in a string like of like the sequenced RNA? Like what do the letters stand for? No, they are split up. No, sequence in the protein. Yes, it's the amino acid and the position that it's in. Those are amino acids. The number is the position in the protein. The letters stand for which amino acid it is. In the number, the letters stand for which amino acids. It is the number somewhere in the sequence. Okay. The amino acid number in the protein. Okay, gotcha. Okay, so then when we say like L455, F486, Q493, what we're saying is that if we were to sequence the spike protein and from start to finish we list out all of the amino acids, these are the like at position 442, we find whatever Y stands for. These are the amino acids that are critical, meaning required for binding to ACE2. Okay, that's what this means, I think. Okay. 5 of these 6 residues differ between SARS-CoV-2 and SARS-CoV. On the basis of structural studies and biochemical experiments, SARS-CoV-2 seems to have an RBD that binds with high affinity to ACE2 from humans, ferrets, cats, and other species with high receptor homology. I'm guessing that means other species with similar type ACE2 receptors. I'm guessing? Okay, I'm guessing. Okay. Don't worry, it's only a 3-page paper. While the analyses above suggest that SARS-CoV-2 may bind... wow, thanks, Freakazoid, for the 1 gifted membership. While the analyses above suggest that SARS-CoV-2 may bind human ACE2 with high affinity, computational analyses predict that the interaction is not ideal and that the RBD sequence is different from those shown in SARS-CoV to be optimal for receptor binding. Okay. So real quick, so if you sequence SARS-CoV, these are the 6 amino acids present at different locations in the spike protein that are essential for binding with the ACE2 receptor in the cell. And then it says that it corresponds to these in SARS-CoV-2. How do you determine what corresponds to what? What does that mean? Like, how can an amino acid in one protein correspond to the amino acid in another protein? Receptor homology would be similarity. Different amino acids at the same position. But they're not in the same position, right? Y442 is at position 442, no? This is at L445 is position 455, no? It's similar functions, basically. Okay. You can look at the whole sequence and do an alignment. There will be conserved domains that line up. When you say line up, what do you mean by that? Like geometrically, or? You can have proteins with different sequences, but similar 3D conformations. Okay. Okay. All right. I don't know if that's ultra super essential. While the analysis above suggests that SARS-CoV-2 may bind human ACE2 with high affinity, computational analyses predict that the interaction is not ideal and that the RBD sequence is different from those shown in SARS-CoV to be optimal for receptor binding. Thus, the high affinity binding of the SARS-CoV-2 spike protein to human ACE2 is most likely the result of natural selection on a human or human-like ACE2 that permits another optimal binding solution to arise. This is strong evidence that SARS-CoV-2 is not the product of purposeful manipulation. Okay, so the so this part of the paper, section 1 here, is arguing that if you were to sequence the spike protein for SARS-CoV, and then you were to run a computational prediction for what a high affinity different protein might look like to bind to the ACE2 receptor, that this sequence of amino acids wouldn't be something that a computer would predict. So the likelihood of this arising from anything but a whole bunch of natural selection, so a whole bunch of viruses infecting and dying and infecting and dying, and then through random chance and mutation, trillions of times or whatever, a new sequence arises that is geometrically and amino acid comparable or capable of binding with ACE2 with high affinity, would be something that a computer just, so in terms of just manually inserting amino acids, which I'm assuming we have the technology to create proteins from chains of amino acids. I'm assuming we can do that. That this would be unlikely to arise from like a computer model, basically. Correct? That's what this section 1 here is speaking about. Correct? Okay. I think. I'm pretty sure. Section 2. Okay. Polybasic furin cleavage site and O-linked glycans. The second notable feature of SARS-CoV-2 is a polybasic cleavage site, RRAR, at the junction of S1 and S2, the two subunits of the spike. This allows effective cleavage by furin and other proteases and has a role in determining viral infectivity and host range. In addition, a leading proline is also inserted at this site in SARS-CoV-2. Thus, the inserted sequence is PRRA. When we're talking about sequences like this, is a sequence of amino acids, right? PRRA, this is an amino acid chain or sequence of amino acids. The turn created by the proline is predicted to result in the addition of O-linked glycans to O-linked glycans to S673, T678, and S686, which flank the cleavage site and are unique to SARS-CoV-2. Like the cleavage site, and okay. Inserted at the site. What is a proline? Senegal. It's also inserted at this site. Thus, the inserted sequence is this turn created by the proline is predicted to result in the addition of proline is an amino acid. Oh, okay. The turn created by the proline. When it says turn, does it mean the morphology, like when this changes shape as a result of the fusion cleavage? Physical turn, okay. And then when the thing turns, because geometry matters or is essential, geometry is an essential function of a protein. So when it turns, as a result of the turning, these three amino acid sites on the S2 portion of the spike protein will pick up O-linked glycans. Correct? Yes, basically. Okay. Okay. Okay, which flank the cleavage site and are unique to SARS-CoV-2. Okay. Proline is an amino acid that tends to induce turns in the protein structure because of its unusual chemical shape. Okay. Okay. Which flank the, okay. Polybasic cleavage sites have not been observed in related lineage B beta coronaviruses, although other human beta coronaviruses, including HKU1, lineage A, have those sites and predicted O-linked glycans. Given the level of genetic variation in the spike, it is likely that SARS-CoV-2-like viruses with partial or full polybasic cleavage sites will be discovered in other species. Genetic variation. It is likely that SARS-CoV-2-like viruses with partial or full polybasic cleavage sites will be discovered in the other species. Sites have not been observed. Okay. Other human beta wait, I'm sorry. What is I don't know what lineage B beta coronavirus means. What does that mean? Lineage B beta coronaviruses. Different strains of the same virus. Okay. Given the level of genetic variation, it's like SARS-CoV-2 like viruses with partial or full polybasic cleavage sites we discovered in other species. Okay. I'm just trying to understand what it's saying here. Or I don't know if anything meaningful or if it matters yet. So this is saying that these particular cleavage sites haven't been observed in related lineage beta, so like beta variants of other coronaviruses, although other human beta coronaviruses, including HKU1, lineage A, have those sites and the predicted O-glycans. Given the level of genetic variation and the spike, it is likely that SARS-CoV-2 like viruses with partial or full polybasic cleavage sites will be discovered in other species. Is this just saying that it's because it exists in humans like this, and there's a lot of genetic variation in the spike protein? If it infects other animals, you're likely to see partial or full polybasic cleavage sites. Okay, I don't think we can care about this yet. Or it's not. Okay. The functional consequence of the polybasic cleavage site in SARS-CoV-2 is unknown, and it will be important to determine its impact on transmissibility and pathogenesis in animal models. Pathogenesis has to do with pattern of it or something, is a process by which a disease or disorder develops. Okay. Animal models. Experiments with SARS-CoV have shown that insertion of a furin cleavage site at the S1-S2 junction enhances cell-cell fusion without affecting viral entry. Okay. Hold on. Experiments with SARS-CoV, so that was the first SARS-CoV virus, the first SARS-CoV infection or virus, sorry. So have shown that insertion of a furin cleavage site, so that means that the part on the S1 or S2 spike protein that would be cleaved by the furin protease, if you insert that site that can be cleaved, it enhances cell-cell fusion, and the cell-cell fusion has to do with what happens at the S2 site where it morphs and then it gets sucked into the wall and the RNA gets injected without affecting viral entry. And okay. I don't know why that wouldn't affect viral entry, but the cell-cell fusion. The cell-cell fusion, what specifically does this refer to? Is it just the change? Is it just the thing where the S2 site causes the shape change, which makes it spit its RNA into the cell? When the membranes combine after the cleavage. Okay, wait, let's look at the viral fusion. Why can't, hello? Viral fusion. Wait, oh my God, does that one girl have a video on that? Viral membrane fusion model. Why do you have two different things? Jesus Christ, principles of viral fusion.
The fusion mechanism can be thought of as...
Oh my God, it's Fauci.
Cleaving the precursor or priming this fusion machinery, localizing the virus to the cell by receptor binding, ultimately by uptake into the endosome. And the triggering of refolding, in the case of flu by low pH, in the case of other viruses, let us say by a receptor or co-receptor binding, that leads to this stereotypical sequence of events. Exposure of the fusion peptide, that's that extended intermediate, insertion of the fusion peptide into the target membrane, and a folding back of the protein that brings together the target and viral membranes.
Okay, sorry, let me just talk through this so far. So cleaving is what happens with furin. Wait, fusion mechanisms. Cleaving is what happens with the furin thing, where it binds to the spike protein, it cuts off the S1 part, and then it goes into the little pocket thing, I forget what it's called, the part of the cell. And then the trigger refolding, that's the S2 part. Once that's activated, the protein refolds itself, and then once it changes shape,
It is that folding back that
then this shit happens, which is what we're looking at right now, I think, right?
overcomes the first of the kinetic barriers. There is a substantial kinetic barrier to squeezing two membranes any closer together than about 10 or 15 angstroms. That is why liposomes, let's say, in solution are stable. Although once fused, they are even more stable. But a liposome preparation doesn't spontaneously fuse because of that kinetic barrier to bringing two bilayers close together. And it is that process that is at least one of the crucial ways in which these proteins facilitate membrane fusion. And they do so by recovering free energy in this foldback process because the primed state is in one way or another metastable. So the fusion of membranes by influenza virus can be thought of then as a triggering process.
This video is not talking about cell-cell fusion, it's talking about virus entry. Cell-cell fusion is an infected cell fused with a neighboring non-infected cell. Oh, well, fuck me. Never mind then. Have shown that insertion of a furin cleavage site at the S1 S2 junction enhances cell-cell fusion without affecting viral entry. Really? How do cells fuse? What does that mean? You searched viral fusion instead of cell-cell fusion. Oh, well, I'm a fucking retard. Cell-cell fusion. Cell-cell fusion of genome-edited cell lines. Ooh, this guy looks like he knows what's up.
This method allows cellular structure and function to be perturbed through the fusion of two different cell types.
Okay.
To address questions such as how do cells respond to changes in organelle number. By imaging fluorescently tagged proteins within fused cells, cellular structures can be referenced back to their cell type of origin to study the mechanisms of organelle homeostasis and function.
For fluorescent dye labeling, see the human cancer cell populations of interest, such that each culture will have expanded to...
Fuck you. Okay, don't care. Never mind. I don't know. Hopefully it doesn't matter. In addition, efficient cleavage of the MERS-CoV spike enables MERS-like coronaviruses from bats to infect human cells. In avian influenza viruses, rapid replication or transmission in highly dense chicken populations selects for the acquisition of polybasic cleavage sites in the hemagglutinin HA protein, which serves a function similar to that of the coronavirus spike protein. Acquisition of polybasic cleavage sites in HA by insertion or recombination converts low pathogenicity avian influenza viruses into highly pathogenic forms. The acquisition of polybasic cleavage sites by HA has also been observed after repeated passage in cell culture or through animals. Does acquisition mean when the amino acid sequence changes or when the RNA sequence changes to have some sequence. You have acquired, is that what we're talking about? The RNA has acquired some set of things that is producing a new sequence that has this particular thing, right? Is that what we mean when we say acquired or acquisition? Yes. Okay. So okay, gotcha. Okay. The function of the predicted O-linked glycans is unclear, but they could create a mucin-like domain that shields epitopes or key residues on the SARS-CoV-2 spike protein. Several viruses utilize mucin-like domains as glycan shields involved immunoevasion, although prediction of O-linked glycosylation is robust. Experimental studies are needed to determine if these sites are used in SARS-CoV-2. Basic furin. Okay. The receptor binding. Okay. So this isn't an argument for evidence or not evidence. These are just features, notable features of the SARS-CoV-2 genome. Okay. Epitopes are where antibodies bind, so if you hide those, the antibodies can't get it. Okay. Whew, okay. Theories of SARS-CoV-2 origins. It is improbable that SARS-CoV-2 emerged through laboratory manipulation of a related SARS-CoV-like coronavirus, as noted above. The RBD, or the receptor binding domain, which is the part that the spike protein uses to connect to ACE2, of SARS-CoV-2 is optimized for binding to human ACE2 with an efficient solution different from those previously predicted. Furthermore, if genetic manipulation had been performed, one of the several reverse genetic systems available for beta coronaviruses would probably have been used. However, the genetic data irrefutably shows that SARS-CoV-2 is not derived from any previously used virus backbone. Instead, we propose two scenarios that can plausibly explain the origin of SARS-CoV-2. One, natural selection in an animal host before zoonotic transfer, or two, natural selection in humans following zoonotic transfer. We also discuss whether selection during passage could have given rise to SARS-CoV-2. Okay. Let me grab a storyline so we can start out. 1 second. Ugh. Okay, we haven't gotten into any of the responses so far, but I imagine that the I imagine that like one response could theoretically be, maybe computer models didn't predict this, but why couldn't scientists in a lab doing gain-of-function research have like naturally stumbled upon this and then decided to make a bioweapon out of it, right? But I don't know if there are any educated people that even respond to these things, or if it's just a bunch of retards, so. Fuck, I can't eat these and read at the same time. Okay. Okay. Theories of SARS-CoV-2 origins. We already did this. Okay, 1. Natural selection in an animal host before zoonotic transfer. As many early cases of COVID-19 were linked to the Huanan market in Wuhan, it is possible that an animal source was present at this location. Given the similarity of SARS-CoV-2 to bat SARS-CoV-like coronaviruses, it is likely that bats serve as reservoir hosts for its progenitor. Although RATG13, things like these I'm guessing are referring to viruses. Although RATG is sampled from a Rhinophilus affinis bat, that's I'm guessing a virus that was infecting a bat, is 96% identical overall to SARS-CoV-2. Its spike diverges in the receptor binding domain, the RBD, which suggests that it may not bind efficiently to human ACE2. Malayan pangolins, Manis javanica, illegally imported into Guangdong province, contain coronaviruses similar to SARS-CoV-2. Although the RATG13 bat virus remains the closest to SARS-CoV-2 across the genome, some pangolin coronaviruses exhibit strong similarity to SARS-CoV-2 in the RBD, including all 6 key RBD residues. This clearly shows that the SARS-CoV-2 spike protein optimized for binding to human-like ACE2 is the result of natural selection. Pangolin coronaviruses exhibit strong similarity to SARS-CoV-2, RBD including all 6 key RBD residues. This clearly shows SARS-CoV-2 spike protein optimized for binding to human-like ACE2 is the result of natural selection. Okay, when we're looking at this thing, nucleotides, fuck, what was this again? Hold on. Molecule that serves as the basic building block of DNA and RNA. Okay. When you, fuck me. When you're talking about like DNA and you're going through the sequence of like DNA, what do you call like adenine, guanine, cytosine? No, it's cytosine, right? And thymine? Something like that. Oh, okay. Okay, never mind. I'm understanding. Okay. The sequences they're nucleotide sequences, but the nucleotides are the same except for the base. The nucleotides is how all these bases connect and make the bullshit. Okay. So for this sequence, there are 29,903. It's RNA, right? So single strand. This is just a sequence of different bases for the nucleotides, different nucleotide bases, or maybe a whole nucleotide is just named for whatever the base is or whatever, right? Okay. So in this whole sequenced string or strand of RNA, the spike protein is coded for here, or encoded for here. So they blow up the whole spike protein sequence here, and then between 300 and 550, this is the receptor binding domain. This is where the receptor is encoded for. Oh, okay, and then this makes sense because, for instance, so if we look up the 6 receptor binding domain amino acids that are, no, wait, nope, never mind. Forget I just said that. No, wait, it is. The spike protein here is broken out into 1285. Okay, all of the key amino acids. So wait, what am I looking at here? So these are nucleotides. This is your. Explain how we go from nucleotide to amino acid. Cynical Brit, where are you? This is a protein. Or no, this encodes for a spike protein. Oh, so this is the spike protein. So this top thing here is the nucleotide sequence of the RNA, and then the nucleotide sequence encodes for this spike protein, and then if you create the spike protein from this sequence, then the spike protein is made of these, is a sequence of these amino acids, and then the critical RBD amino acids for binding are, we can find them here, right? Okay, so if we're looking at so up here in this paper, it brought out with corresponding to, and SARS-CoV-2, so like L455, so 455, 486, 493, so from 455 to 505, we would expect to see that in here, right? Because this is the RBD, the receptor binding domain, and this is where the most important ones are, or the essential ones for binding to ACE2. Okay. And it looks like that might even be what's like highlighted here, I guess. Okay. The S1 subunit is the thing that gets cleaved by the furin entering the cell, by the protease entering the cell, and then the S2 one is the one that causes it all to get erect and change shape and all that. These are the key amino acids that are present. Okay, so what is this? So we have receptor binding domain ACE2 contact residues. Human, SARS-CoV-2, bat, rat G13, pangolin. Okay. What exactly is this shit? What are we looking at here? Is this looking at similar binding, receptor binding domain amino acids in these viruses and if they line up with these basically? I think so. So these are the 6 key RBD amino acids, and then this is the amino acids that are the reference, I'm guessing, to the binding amino acids here found in these other 6 viruses. Yes, okay. Okay, and then the polybasic cleavage site, which is the S2 thing, that when it gets interacted with, it changes shape, and then because of the shape change, the introduction of some amino acid changes the shape in such a way that the surrounding amino acid sites absorb hydrogen ions, H+, as the shape change. Because of the shape change, it lets things bind around the cleavage site, I think, right? Okay, and then these don't have correspondence in the other viruses. Is that what this means here? Maybe? Okay. Figure 1, features of the spike protein in human SARS-CoV-2 and related coronaviruses. A, mutations in contact residues of the SARS-CoV-2 spike protein. The spike protein of SARS-CoV-2, red bar at the top, was aligned against the most closely related SARS-CoV-like coronaviruses and SARS-CoV itself. Key residues in the spike protein. What does a residue mean here? Somebody explain that? It's the R group of an amino acid, basically. What's the R group of an amino acid? A residue is an amino acid in a protein. Oh, the residue is what makes each amino acid unique. Oh, okay. Okay. Key residues in the spike protein that make contact with ACE2 receptor are marked with blue boxes of both SARS-CoV-2 and related viruses, including SARS-CoV Urbani strain. Acquisition of polybasic cleavage site and O-linked ligands, both the polybasic cleavage site and the 3 adjacent predicted O-linked ligands are unique to SARS-CoV-2 and were not previously seen in lineage B beta coronaviruses. Sequences shown are from the NCBI GenBank. Accession codes. The pangolin coronavirus sequence or a consensus generated from. Okay. Although the rat G13 bat virus remains a cluster of SARS-CoV-2 across the genome, some pangolin coronaviruses exhibit strong similarity to SARS-CoV-2 in the RBD, including all 6 key RBD residues. This clearly shows that the SARS-CoV-2 spike protein optimized for binding to human-like ACE2 is the result of natural selection. Okay. Okay. Cynical guy. Okay. Thomas. Okay. So, is the argument basically, just so I'm understanding up to this point. So if you were to run computer models on SARS-CoV, and then you use what we consider to be key amino acids and shapes for binding to the ACE2 receptor, and you were to try to predict other known efficient or effective ways to combine with ACE2, you would not predict this particular sequence. It hasn't been predicted yet, or a computer model wouldn't predict it, which makes it more likely that it was a result of natural selection. My counter argument to that would have been, okay, well, maybe they through some sort of gain of function research, maybe they discovered this or something like that, right? But then it seems like it's brought up here. So I know they haven't put strong arguments yet, but it seems like a counter argument that, well, in all of these other unrelated viruses, or not, I shouldn't say unrelated, I'm sorry. In these closely related viruses that are infecting other creatures, this same sequence of amino acids or the same structure kind of or functionality from these has emerged as well. So you would have to try to explain that. In order for this to be possible, the human virus would have had to have escaped and infected all of these creatures as well, because this particular RBD amino acid sequence, I don't know if sequence is the right word here, is thus far unrecorded or unknown. And it seems to have independently evolved in these different strains of closely related viruses. So this configuration, whatever, right? Okay. Okay. And then they're also making the prediction, I guess, although I don't know if it's relevant to it being human-made or not, that because of the variability of the spike protein, it's likely that in B lineage evolutions or whatever, or mutations of these viruses, that you would expect to see polybasic cleavage sites emerge, even if they're not there yet. Correct? That's like a prediction they're making about evolutions of these other viruses. Where did my chemistry guy go? How dare you not Okay, I'm going to assume I'm correct. Okay. Neither the bat beta coronaviruses nor the pangolin beta coronaviruses sampled thus far have polybasic cleavage sites. Oh, okay. Although no animal coronavirus has been identified that is sufficiently similar to serve as the direct progenitor of SARS-CoV-2, the diversity of coronaviruses in bats and other species is massively undersampled. Mutations, insertions, and deletions can occur near the S1, S2 junction of coronaviruses, which shows that the polybasic cleavage site can arise by a natural evolutionary process for a precursor virus to acquire both the polybasic cleavage site and mutations in the spike protein suitable for binding to human ACE2. An animal host would probably have to have a high population density to allow natural selection to proceed efficiently, and an ACE2 encoding gene that is similar to the human ortholog. Gotcha. Okay. Okay, I saw Wang Sima contention. That name looks like a troll, I'm going to be honest, but okay. I read your message. Thank you. Okay. Gain-of-function research could stumble on the sequence, but it's very unlikely. Usually the way that kind of research works is creating random sequences, picking the best one somehow, and repeating it over and over again. It's much more likely for natural selection to do it, and it can... Okay, sure. Okay. That's okay. All right. We're still just understanding the paper. I don't have any strong positions yet right now. Okay. All right. 2. Natural selection in humans following zoonotic transfer. It is possible that a progenitor of SARS-CoV-2 jumped into humans, acquiring the genomic features described above through adaptation during undetected human-to-human transmission. Once acquired, these adaptations would enable the pandemic to take off and produce a sufficiently large cluster of cases to trigger the surveillance system that detected it. All SARS-CoV-2 genomes sequenced so far have the genomic features described above and are thus derived from a common ancestor that had them too. The presence in pangolins of an RBD very similar to that of SARS-CoV-2 means that we can infer this was also probably in the virus that jumped to humans. This leaves the insertion of polybasic cleavage sites to occur during human-to-human transmission. Okay. Estimates of the timing of the most recent common ancestor of SARS-CoV-2 made with current sequence data point to emergence of the virus in late November 2019 to early December 2019, compatible with the earliest retrospectively confirmed cases. Hence, this scenario presumes a period of unrecognized transmission in humans between the initial zoonotic event and the acquisition of the polybasic cleavage site. Sufficient opportunity could have arisen if there had been many prior zoonotic events that produced short chains of human-to-human transmission over an extended period. This is essentially the situation for MERS-CoV, for which all human cases are the result of repeated jumps of the virus from dromedary camels, producing single infections or short transmission chains that eventually resolve with no adaptation to sustained transmissions. Studies of banked human samples could provide information on whether such cryptic spread has occurred. Retrospective serological studies could also be informative. What is a is that like serializing or studying like a sequence? What does serological mean? Serology in general refers to the scientific study of blood serum and other bodily fluids. Okay, that's what your mom does. Ha, woo yeah! And a few such studies have been conducted showing low-level exposures to SARS-CoV-like coronaviruses in certain areas of China. Critically, however, these studies could not have distinguished whether exposures were due to prior infections with SARS-CoV, SARS-CoV-2, or other SARS-CoV-like coronaviruses. Further serological studies should be conducted to determine the extent of prior human exposure to SARS-CoV-2. Selection during passage. Basic research involving passage of bat SARS-CoV-like coronaviruses in cell culture and or animal models has been ongoing for many years in biosafety level 2 laboratories across the world, and there are documented instances of laboratory escapes of SARS-CoV. We must therefore examine the possibility of an inadvertent laboratory release of SARS-CoV-2. In theory, it is possible that SARS-CoV-2 acquired RBD mutations during adaptation to passage in cell culture, as has been observed in studies of SARS-CoV. The findings of SARS-CoV-like coronaviruses from pangolins with nearly identical RBDs, however, provides a much stronger and more parsimonious explanation of how SARS-CoV-2 acquired these via recombination or mutation. It's possible that SARS-CoV mutations during adaptation to passage in cell cultures, as has been observed in studies of SARS-CoV-like coronaviruses from pangolins with nearly identical RBDs. Oh, okay, that's the other argument I was making. Okay, however, provides a much stronger and more parsimonious explanation of how SARS-CoV acquired these. Okay, the acquisition of both the polybasic cleavage site and predicted O-linked glycans also argues against culture-based scenarios. New polybasic cleavage sites have been observed only after prolonged passage of low pathogenicity avian influenza virus in vitro or in vivo. Prolonged passage. Okay. Furthermore, a hypothetical generation of SARS-CoV-2 by cell culture or animal passage would have required prior isolation of a progenitor virus with very high genetic similarity, which has not been described. Subsequent generation of a polybasic cleavage site would have then required repeated passage in cell culture or animals with ACE2 receptors similar to those of humans, but such work has also not previously been described. Finally, the generation of the predicted O-linked glycans is also unlikely to have occurred due to cell culture passage, as such features suggest the involvement of an immune system. Okay, so this is saying that polybasic cleavage sites are not something that generally just happen like instantly from one mutation to the next, but in general these occur after many generations of viral transfer, and they generally require polybasic cleavage sites generally develop after many transfers when there is a presence of some immune system natural selective mechanism. And so the chances of a polybasic cleavage site developing on in this strand of RNA, the acquisition of that polybasic cleavage site would probably be highly unlikely in a laboratory where they haven't described. When they say described, they mean published research on or illustrated a sequence of, like published a sequence of this RNA. It would be highly unlikely for that to emerge instantly. It would have to be done through many generations. And then the fact that the RBD has some particular amino acids that also aren't predicted by a computer, but have developed in parallel in other animals in the wilderness suggest some common ancestor, I think. Okay. So the zoonotic transfer is essentially saying there's some common ancestor here, or maybe the pangolin one jumped from bat to human, and then there was a bunch of transmission from human to human without much disease feature. And then as that transmission occurred, eventually the polybasic site was acquired because an immune system will select for polybasic cleavage sites, and then once that happened, it basically became COVID-19. So okay. Basically, they would need to find. Okay, all right. We're almost done. I think we're on the last page. Okay. No, we're not. Sorry, we're almost done with page 2. All right. Oh, no, wait. We're on page 3. Last page, okay. Conclusions. In the midst of the global COVID-19 public health emergency, it is reasonable to wonder why the origins of the pandemic matter. Detailed understanding of how an animal virus jumped species boundaries to infect humans so productively will help in the prevention of future zoonotic events. For example, if SARS-CoV-2 pre-adapted in another animal species, then there is the risk of future reemergence events. In contrast, if the adaptive process occurred in humans, then even if repeated zoonotic transfers occur, they are unlikely to take off without the same series of mutations. In addition, identifying the closest viral relatives of SARS-CoV-2 circulating in animals will greatly assist studies of viral function. Indeed, the availability of the RATG13 bat sequence helped reveal key RBD mutations in the polybasic cleavage site. The genomic features described here may explain in part the infectiousness and transmissibility of SARS-CoV-2 in humans. Although the evidence shows that SARS-CoV-2 is not a purposefully manipulated virus, it is currently impossible to prove or disprove the other theories of its origin described here. However, since we observed all notable SARS-CoV-2 features, including the optimized RBD and polybasic cleavage site, in related coronaviruses in nature, we do not believe that any type of laboratory-based scenario is plausible. More scientific data could swing the balance of evidence to favor one hypothesis over another. Obtaining related viral sequences from animal sources would be the most definitive way of revealing viral origins. For example, a future observation of an intermediate or fully formed polybasic cleavage site in a SARS-CoV-2-like virus from animals would lend even further support to the natural selection hypothesis. It would also be helpful to obtain more genetic and functional data about SARS-CoV-2, including animal studies. The identification of a potential intermediate host of SARS-CoV-2, as well as sequencing of the virus from very early cases, would similarly be highly informative. Irrespective of the exact mechanism by which SARS-CoV-2 originated via natural selection, the ongoing surveillance of pneumonia in humans and other animals is clearly of the utmost importance. Okay. Jesus. Okay, so when this paper was published, the argument was the researchers in this early paper argue that parallel evolution, I shouldn't say parallel evolution, right? I should say that parallel demonstrations of 6 key amino acids in the RBD was binding domain, R binding domain. What was it? Receptor binding domain. Demonstrate 6 key amino acids. Being present in other similar hosts in Wuhan in a configuration that was not predicted to be effective for infection for binding via ACE2. Infection is not predicted to be effective for infection. Suggest a common ancestor for the virus. And then they also argue that the evolution of, or the, I should say acquisition is probably more right. So argue that the acquisition of a polybasic cleavage site suggests natural selection occurred over many generations within the presence of an active immune system, which is unlikely to happen in a laboratory setting, especially with no evidence or documentation of any attempt with a particular, sequence? Nucleotide sequence virus to do so. Okay. 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 This was the first big paper that was authored when the virus first dropped. This was a few months after. This was published in what was the date? Like March of 2020, I think. April of 2020. Okay. Jesus Christ. April 2020. Okay, that's good enough. Okay, all right. We don't need this paper. Okay, that was the first paper. Back to the debate. Session 1, $100,000 debate. Oh boy. Please don't be
Hi,
shitty
everyone.
audio.
My name is Will Van Truren, and I will be participating and moderating in a debate on the origins of COVID. I'm going to call the order to session. Everyone's here. First, we'll have the judges read affirmations indicating neutrality and lack of conflict of interest. We'll have the participants introduce themselves, and then we will be having two 90-minute initial opening statements from each side, followed by rebuttals from each side and questions from the judges. So to read my oral affirmation.
Last mention, but the emails of the scientists were published and they were not as confident in this conclusion. For what? Hold on. Scientists discuss masking their involvement in key journal letter on COVID origins emails shows. Or emails show. EcoHealth Alliance President Peter Daszak, the head of an organization involved in research that genetically manipulates coronaviruses, discussed hiding his role in a statement published last year in The Lancet that condemned as conspiracy theories concerns that the COVID-19 virus may have originated in a research lab, emails obtained by U.S. Right to Know show. The Lancet statement signed by 27 prominent scientists have been influential in tamping down suspicions. Drafted the statement and circulated it, but the emails reveal. Okay. Let's see. From Peter Daszak. I spoke with Lympha last night about the statement we sent around. He thinks, and I agree with him, that you, me, and him should not sign this statement, so it has some distance from us and therefore it doesn't work in a counterproductive way. Jim Hughes, Linda Safe, Hume Field, and I believe Rita Colwell will sign it, and then I'll send it around to some other key people tonight. We'll then put it out in a way that doesn't link back to our collaboration, so we maximize an independent voice. I also think this is a good decision, otherwise it looks self-serving and we lose impact. So these guys are saying that they shouldn't sign this public statement because they felt like because they're involved in organizations that do gain-of-function research, so it would look bad to have I mean, this seems reasonable, no? Like, yeah, like people would probably attack you if you, or would say that like, oh, these guys are just signing it because it benefits their companies. But the emails revealed that these thought they should not sign the statement, so the master involvement in it. Leaving their names off the statement would give it some distance. Doesn't know that he could sell it around or send it around to other scientists to sign. We'll then put it out in a way that doesn't link back to our collaboration, so maximize independent voice. He wrote, the two scientists Daszak wrote to about the need to make the paper appear independent of EcoHealth are coronavirus experts Ralph Baric and Lympha Wang. In the emails, Baric agreed with the exception of silent statement, writing otherwise like self-serving, will lose impact. He did ultimately sign the statement himself, but he was not identified as its lead author or coordinator of the effort. The emails are part of a tranche of documents seen by U.S. Right to Know that showed it last year to undermine the hypothesis that SARS-CoV-2 may have leaked from the One Institute. Hold on. Who linked this initially? Link me something that says what you said in chat. Otherwise, you're permabanned. You said that there was a statement, or that emails showed by them showed that they were not confident about the paper. This just sounds like it has to do with them signing a PR statement. Who linked this initially? Hassan was on Twitch threatening a chat with a gun. Stephen IRL, a bounty hunter got permit. Or a bounty hunter got permit. No warning from Twitch for brandishing a firearm when entering a house for his safety. Stephen need to, or I'm sorry, Jesus. People need to get onto Twitch. Okay. Sorry, I was looking at links too hard. We'll search for the proper link. You linked this like 10 times, didn't you? There's also some evidence in the emails of these researchers in regards to their competence and their claims. Last message from Miller's paper, but the emails of the scientists were published and they were not as confident in this conclusion. Okay. You survived for now. I don't know how you linked this 4 times and you didn't read it, but okay.